The overall goal of this procedure is to demonstrate a system for generating unmarked mutants in the Cyanobacteria Synechocystis species PCC6803 and marked mutants in Synechococcus species PCC7002.
This method can help answer key questions in the cyanobacterial genetics field, such as how to introduce chromosomal alterations into a strain via insertion of an antibiotic resistance gene, followed by subsequent removal of this cassette using a negative selectable marker. The main advantage of this technique is that strains can be repeatedly genetically manipulated, allowing as many alterations to be introduced into a strain as desired.
After preparing media, cyanobacteria strains, and plasmids according to the text protocol, set up a fresh culture by inoculating a loop full of cyanobacteria cells into 30 to 50 milliliters of BG11 medium. Grow the culture in the light at 30 degrees Celsius for 2 to 3 days to an OD 750 equal to 0.2 to 0.6. Following the incubation, centrifuge 1 to 2 milliliters of the culture at 2,300 Gs for 5 minutes. Then, after discarding the supernatant, use BG11 medium to wash the pellets once, gently pipetting to resuspend the cells, as vortexing may result in the loss of pili, which are essential for DNA uptake. After pelleting the cells again and removing the supernatant, add BG11